Assalamualaikum everyone. Welcome to Biology online class. This is Salmakta Sweetie. In this video, we are going to learn about genetic engineering, a very advanced and modern technology. At first, let's know what is genetic engineering. Genetic engineering is the engineering of genes. You have already heard about gene. Gene is a segment of DNA that carries a character. For example, in this picture, this is a long DNA strand, and this segment or part of DNA is a gene, and it can carry a character, carry a character. For example, our skin color or our growth or our eye color or any kind of character of any living organisms. And genetic engineering is the technique of transferring a gene from one organism to another organism. As we know that gene carry character, then if we transfer a gene from one organism to another, actually we are transferring a new characteristics to an organism. And all the techniques involving this transferring DNA is commonly called recombinant DNA technology or rDNA technology. And recombinant DNA technologies involves a series of procedures used to recombine DNA segments. As we collect one DNA segment from one organism and we join the segments to another organism's DNA, actually we are recombining different DNA segments from different organisms, and that's why it is called recombinant DNA technology. And this transfer of genes can be done from bacteria to human plants or human plants to bacteria, and it can be also from animals to plants or plants to animals. And the organism that is produced by genetic engineering with a new characteristic is called genetically modified organism or genetically engineered or transgenic organism. The term genetic engineering was first coined by Jack Williamson in his science fiction novel Dragon's Island, published in 1951. And in 1973, Herbert Boyer and Stanley Cohen first created a transgenic organism by inserting antibiotic resistance genes into the plasmid of an Escherichia coli bacteria. If we want to learn recombinant DNA technology, we at first we have to know some terms, and that is, first one is the plasmid. We will hear about plasmid in these procedures. That's why we have to know what is plasmid and what is the function of the plasmid. So here you are seeing this is a bacteria cell or a prokaryotic cell, and plasmids are defined as extra chromosomal DNA. Why it is extra chromosomal? Because this is not is a part of the main chromosome of the bacteria. That's why it is called extra chromosomal bacteria. Here you are seeing this is bacterial DNA, and this bacterial DNA is responsible for all the living characteristics of the bacteria. And these are plasmids. And that's here plasmids are not is a part of the bacterial DNA, and that's why it is called extra chromosomal DNA. And self-replicating properties. It have self-replicating properties, that is, it can replicate or it can copy itself without any kind of help from the bacterial DNA. That's why it is called self-replicating properties. And these plasmids are not necessary for the survival of the bacteria. As this is extra DNA, and DNA always carries characters, so these plasmid DNA give extra advantageous characteristics to the bacteria that holds the plasmids. Plasmids are usually circular in shape, and one cell can have more than one plasmid. Here you are seeing 3 plasmids in a bacteria cell. Plasmids are called vector in this recombinant DNA technology. Vectors, that means a carrier, and plasmids carry, maintain and replicate cloned gene that we want to transfer. Which gene we want to transfer, that will be cloned, and plasmids carry, maintain and replicate the gene inside a host cell. That's all about plasmids, and we have to know about restriction enzyme, because this is a very important part in the recombinant DNA technology. Restriction enzyme is a special type of enzyme found in bacteria or prokaryotic cells, and it is called a molecular scissor because it can cut DNA at a specific base sequence in DNA. It can recognize a particular base sequence. For example, here you can see eco R1. This is a name of a restriction enzyme, and it can cut at base sequence GAATTC. You have already know about base sequences, that is, these are guanine, adenine, thymine and cytosine. And when these base sequences arise in a DNA, eco R1 enzyme can recognize this sequence, and it can cut the bond inside these sequences, and it cut in this pathway that is shown in the figure. And by cutting here, 2 portion of DNA has been created, and the end of the DNA is complementary and can bind to each other. You can see A to T, A to T, A to T, T to A and T to A, and such like they can bind to each other. If we cut numerous DNA molecule by one specific enzyme, that will produce end always like this, always same end produce, and they can bind to each other. Let's know the steps in the recombinant DNA technology. Step 1 is isolation. So the target DNA or gene to be cloned, or the gene that we want to transfer from one organism to another organism, first must be isolated from that organism having that. For example, here it's a human cell. You can imagine about insulin gene, and it's the nucleus, and inside the nucleus there is DNA. And so at first we have to isolate the gene of the human cell, and then we have to isolate a plasmid vector from a bacteria, because we have to transfer the gene of interest from here to a plasmid. What is gene of interest? A gene of interest is a fragment of gene whose product, gene's product, we already know that a protein or enzyme or hormone, everything is a protein that interest us, and that gene we want to transfer from one organism to another organism, and that character we want to transfer from one organism to another organism. For example, here we have counted gene encoding for the hormone insulin, and here is the portion in the human genome where gene for insulin is located. After the isolation of the DNA and plasmid, we have to cut the plasmid and the DNA and the cell's DNA with same restriction enzyme. We use same restriction enzyme because same restriction enzyme will produce complementary ends. Complementary ends that can bond form to each other. Restriction enzyme cut both of them at specific region. As bacterial plasmid is so small, that's why it have only one region to cut. But in the human DNA or in the animals DNA or in the plants DNA, there is many region, and it will be cut into many segments, from which one segment will have the gene of interest, and the ends that are produced is called sticky ends because they can stick together or join together. After cutting, the segments are joined to the plasmid. That is, this gene of interest will be transferred or joined to this plasmid. How can we do it? We do it by mixturing the donor DNA. Donor DNA is called the gene of interest, is called the donor DNA fragment, and plasmid vector are mixed together so that they will join together by forming bond at the complementary ends. And here you are seeing two ends form bond together and they are joined to each other. But in this figure you are seeing there is a gap, and we have to fulfill this gap. This gap is due to a lack of bond. That is, there is no bond between the segments in this region. That's why there is a gap. And in the next step, we are going to join the gap. And this step is called ligation. Ligation is performed by an enzyme that is called DNA ligase. So the recombined DNA is treated with DNA ligase, and base pairing of donor DNA fragment and plasmid vector occurs. The resulting DNA molecule, here the bond will form here, and this is the result of the bond formation, and this molecule is a hybrid of two molecule, that is one molecule and this is the another molecule. So in the terminology of genetics, this intermixing of different DNA strands is called recombination, and that's from the name came from. And these DNA molecule that have arisen from the ligation or from joining is called recombinant DNA plasmid. So what is the role of DNA ligase? DNA ligase, here you are seeing this figure will clear you. There is a phosphodiester bond is missing, and DNA ligase synthesize this bond here and repair the gap. This is the function of DNA ligase enzyme. Next, step 5 is the insertion. We have already formed recombinant DNA, and we have to insert it to a living organism. That is, we most of the time we choose a bacterium. And so insertion is the injection or transferring the recombinant DNA molecule to a host cell. Here bacteria is the host cell, which will carry the plasmid or recombinant DNA plasmid, and which will give it a living character. Then the step 6 is the cloning. Cloning is the production of exact copies of a particular gene. That is, we have transferred the gene or recombinant DNA plasmid into the bacterium, and we have to give it nutrients or medium to grow, and if we give a nutrition to the bacterium, and that is recombinant, or this bacteria now can be called transformed organism or transgenic bacteria, and this bacteria, if we give nutrients for growing, it will divide or it will multiply its number, and when it divides, it will make copies of its own DNA along with the plasmid DNA with new characters, and the bacterium can divide in a very short time, and that's why we are using bacteria, and bacteria will grow to numerous many cells, and each of the cells will carry the recombinant plasmid, and in the recombinant plasmid there is the DNA we have inserted, and it will carry the character we have inserted, and that's why in a very short time millions copies of the gene will be formed by cloning. And this is the final goal for the genetic engineering technique. We want to produce our desired gene in a huge amount. So let's in a summary we will know the steps in the recombinant DNA technology. First one, we will isolate the desired gene and the plasmid gene. We'll isolate the desired DNA and the plasmid, and then the DNA, both of the DNA will be cut with the same restriction enzyme. Then the pieces of the DNA will be joined, and the recombinant DNA plasmid will be inserted into a host organism. Then the recombinant organism will be selected from the non-recombinant organism by specific methods, and the recombinant organism will grown in nutrients, and finally we will separate our products by various methods. This genetic engineering technique is a very modern technology, but it have many applications in our daily life. The most important one is in agriculture. In agriculture we use genetic engineering for developing crops, for example insect resistant crop for that can kill the insect, or that are not attacked by the insect or virus, or it we can produce herbicide tolerant crops. We use various herbicide for our farming, and those herbicides can harm our crops. So if our crops are tolerant to the herbicide, then it will be very advantageous for us, and we can form salinity, that is high salt concentration, and drought, that is dry condition resistant crops that can grow in these conditions, and we can enhance the nutritional quality of any kind of food. For example, vitamin A enriched rice has been developed. If we intake or eat the vitamin A enriched rice, we don't have to intake vitamin A from outer source. And we can make seasonal crops available round the year. Other applications include we can genetically engineered animals to increase growth of animals, or growth, for example growth hormone gene is transferred here to increase the growth of any animals, to increase the amount of meat, or we can increase the amount and quality of milk and fur. In healthcare, we can use genetic engineering to develop drugs that different necessary proteins such as hormones, for example insulin or growth hormones, that can be used to treat different diseases, for example diabetes or dwarfism. And in environment, we can use genetically modified organism to the management of industrial waste and sewage, because they have the capability to degrade the waste products that are released in the environment and which are polluting in our environment. So this is all about genetic engineering. Hope everyone have cleared. This is all for today. Thank you all. Allah Hafiz.